supervisor were Rachel Rupp and Nick Friggans. I did it in Toulouse, in INRA. So the title of my PhD was combining genetics and modeling approaches to develop new non-invasive biomarkers of resilience based on milk metabolites. So firstly, what is resilience? So resilience can be defined as the capacity of an animal to overcome short-term environmental disturbances and then to quickly return to the pre-disturbance state afterward. So selecting for more resilient animal could be of increasing value, considering the increasing number and intensity of the environmental disturbances we are overcoming. So the hypothesis we based this study on was that there is diversity of metabolic strategy to respond to an underfeeding challenge, and these strategies are part of the heritable resilience mechanism in dairy goats. So if we want to study dairy goats' resilience, we firstly need an environmental perturbation, so the goats will be able to express their resilience. And we also need inter-individual variability concerning resilience. Is to say we need goats that are more or less resilient. We also need to grasp the profile on which they will adapt to the perturbations. We need longitudinal recording, and considering the complexity of the resilience adaptability, we also need, it can be useful to have multivariate data to understand this complex process. And finally, once we have goats, we respond differently to a given perturbation, we need a validation measure to say, okay, this goat is better. This way of responding to a challenge is better. And we actually used functional longevity as a validation proxy. So functional longevity is defined as the true longevity of a goat, is to say its lifespan, corrected for productivity. And actually, we can consider that all other things being equal, a goat with a better resilience will live longer because it will overcome better all the accumulated perturbation it will face through life. And we actually also used functional longevity as a way to create inter-individual variability in our animal model. Actually, we run a divergent selection for functional longevity in INRA. So let me explain that. We actually created two divergent lines for functional longevity. So there was a high longevity line and a low longevity line. So based on the national performance recording for dairy goats, we ran a genetic evaluation for all the artificial insemination bucks, and we estimated a breeding value for functional longevity for all those bucks based on the survival of their daughter. And based on this evaluation, we selected the best and the worst bucks of reds. And then we mated these bucks with the goats we had in our experimental facility. And it created the low and the high longevity lines. So this selection was made in two experimental facilities of INRA, one in Mozart in Paris and one in Bourges. In total, we bred more than 500 goats of the two longevity lines. And then we wanted these goats to express their resilience mechanism. So we submitted our goats to a 2-day underfeeding challenge, meaning the beginning of the first lactation. And so during this underfeeding challenge, we only gave straw to them. and water, obviously. And for every day before, during, and after the challenge, we sampled blood and milk daily. And in the sample, we measure several milk metabolites. So in the milk, we measured 14 milk metabolites. So these milk metabolites describe several metabolic pathways. Some were involved in more intracellular energetic pathway, like glucose, glucose 6-phosphate. Other reflected more the protein metabolism, such as urea, UFAT, glutamate. There was also metabolites linked with milk fat content, such as triacylglycerol, cholesterol, and choline. There was also the VGB, which is a ketone body which reflects the energetic metabolism of animals, and the LDH, which is an enzyme, and it's linked with inflammation of the mammary. Can be used to detect the mastitis. We also had other milk records, such as milk yield, fat content, protein content, and somatic cell score. And we also measured 4 blood metabolites: glucose, urea, BHB, and NIF. In this presentation today, I will not describe much the blood metabolites, but only at the end for the genetic analysis. So the interest of the milk metabolites is that they reflect several metabolic pathways, firstly. Then they are very easily sampled, which is can be of great value if you want to implement on-farm selection for resilience for goats. And also, compared with blood metabolites, they fluctuate less across the hour of the day. The glucose, for example, in the blood can vary much from 1 hour to the next one. But since the glucose in the milk accumulated between 2 milkings, there is less fluctuation. So my presentation today is in 3 parts. So firstly, we will validate the animal model by comparing the survival of the high and the low longevity line. And we will ask what other performance is different between these goats. Secondly, we will study the link between resilience and energetic metabolism based on the milk metabolites samples during the underfeeding challenge. And finally, we run a genetic analysis of the metabolic profiles during the underfeeding challenge. So firstly, the validation of the animal models. So we compared the 2 longevity lines. In this part, we will focus only in one experimental facility, Bourges, because there no culling for performance reason occurred. So we could have a clear assessment of the functional longevity of those goats in this farm. That was not the case in the other farm. We also had some health, growth, dairy performance recording in this farm. So firstly, we compared the survival of those goats, and we show that indeed the high longevity line had a better survival than the low longevity line in red. And this difference occurred mainly after the 2 years of age of the goats. We also showed that the high longevity line was heavier during the beginning of the first lactation, had wider chest for the same height, indicating that the high longevity line were more fatty. And there was also a lower milk fat protein ratio among the high longevity goats, indicating that a high longevity goat had lower body fat mobilization during the beginning of the first lactation. And finally, we also had lower somatic cell score among the high longevity line, indicating a better mammary health. We did not show any difference concerning the health treatment recording, the fertility or culling reason among profiles. So as a conclusion, we the genetic selection we ran created indeed two lines with different survival. And the high longevity line seemed to have lower body fat mobilization during the beginning of the first lactation and better mammary health. So we indeed have animals with contrasted levels of resilience that now we can study. So this goat was submitted to this 2 days underfeeding challenge, and we measured the 14 milk metabolites before, during, and after this challenge. So each panel corresponds to milk metabolites. And the green area here corresponds to the 2-day underfeeding challenge. So you can see that all the metabolites vary during this challenge. Each line is a goat, and the color of the line corresponds to the facility and the year where it was conducted. We can see also that there is year form effects, especially here for urea, for example. You can see that in Mozart, the urea level was lower. But even within each year, you can see that there is inter-individual variability, and not all of the goats respond the same to this challenge. So this is the variability we want to study. And we wanted to model. So firstly, we had a choice in the model between the flexibility and the interpretability of the model. Here we had few hypotheses concerning the shape of the profile of the milk metabolites. So we wanted to have a quite flexible model. And then we did not want only to focus on each milk metabolite profile separately, but we wanted to have an overall, to grasp the overall metabolic response. So for this part of my PhD, I worked with Professor Jigo Tao from the Simon Fraser University in Vancouver. I've been there for 4 months in the Department of Statistics, and we worked on a functional data analysis approach. So the first step of this approach was the spline interpolation. We used a natural cubic spline. So here it's the data for one goat, for all the metabolites. The dots are the real data, and you can see the line is the smoothing spline. So firstly, here I'm going to present the data for isocitrate. So that's all on the left, you've got all the goats. The blue goat is just, the blue line is one goat that we aligned. So it's before and after the smoothing. And then we had the problem of the farm year effect we wanted to correct for. We used the functional linear regression in order to correct for this effect. So just like normal linear regression, we estimated an intercept and the year effect, but here they are curves. And by removing the year effect, you've got an estimation of each individual profile corrected for year effect. So here on the left, you've got the mean curve before this correction. On the right, after it's the same. And then it leads us to the part of our modeling. We use the functional PCA to model our profile. So we run a functional PCA for each metabolite. So here is the example for isocitrate. Actually, a functional PCA will do a little bit like a PCA. So it will decompose the overall variability of our group of curve into several principal components. But here, the 3 components are curves. You can see the number of components and the shape of components are automatically defined to represent at least 90% of the variability. And once you've got those principal components, you can actually calculate the individual functional PC scores for each goat that will describe the profile of those goats. So actually you multiply the components with your individual curves like that. And then you calculate the area under curve and you've got your score for this goat. And you can understand, based on these scores, you can understand the shape of the profile of these goats by looking at the shape of your principal components. So, for example, the principal component 1 here is always positive and more or less flat, let's say. So this component will describe the overall mean level for isocitrate. You see this goat has a score of 1 isocitrate, which is low, minus 129. So it means that this goat has an overall concentration of isocitrate which is low, and we can see that this goat has a low concentration of isocitrate. For the second component, it's positive before and after the challenge, but negative during the challenge. So you can say that because, again, this goat has a low score for 2 isocitrates, this goat has a low peak of isocitrate during the challenge, which is again true. So you do it for each metabolite, and you've got your 48 functional PC scores that describe the overall metabolic response to the underfeeding challenge. And firstly, we wanted to compare this metabolic response between the high and the low longevity line. So we run supervised clustering, a sparse partially square experiment analysis, to predict the genetic line of the goat based on the 48 scores. And it happened that we could not predict it. And the balance error rate was super high. It was almost exactly random. So the two longevity lines did not show a different metabolic reaction to the underfeeding challenge. So we decided not to stop there and to consider that the high and low longevity line were just a way to increase the overall variability of our animal samples and to explore this variability. So we ran non-supervised clustering, and it showed us that there was a cluster of goats with different metabolic response to the underfeeding challenge. So there was 36, 53, and 49 goats respectively in the three clusters. And when we compared the survival of those three clusters of goats, we saw that indeed the cluster 1 in dark purple had significantly lower survival than the two other clusters. And what is this cluster 1? When we look at the mean metabolic curve of these three clusters, we can see that the cluster 1, again in dark purple, has actually a high body fat mobilization during the underfeeding challenge, with a high increase of BHB, cholesterol, choline, and triglycerides. Sorry, I did not say that the red area corresponds to where the curves are significantly different. And then the cluster 1, again, had a sharper decrease and slower increase of the milk, sugar, and protein-related metabolism. So it's glucose, glucosylphosphate, glutamate. You see it's lower for the cluster 1. And also for glutamate, NHE, and triglycerides, NH2. And we could see it was not as strong, but we could see that the LDH was higher for the cluster 1 after the challenge. So the inflammation linked to the challenge seemed to be more persistent. So the conclusion of this part was that we developed a nice way to deal with this data. There was no difference of metabolic response to the challenge between lines. but a high metabolic response to the underfeeding challenge, which is the cluster 1, is also associated with poor survival. And this shows a link between energetic metabolism and resilience. So finally, we ran a genetic analysis on the metabolite profiles. So firstly, we ran an heritability estimate for all the functional PC scores for the milk metabolite profiles, but also for the blood metabolite profiles. For the blood, we had more goats. And those goats were also genotypes, but only a part of those goats so far. So we had 150 goats who had genotypes for and blood metabolite profiles, and not many goats for the milk. So we ran a GWAS only on the blood metabolite profile. And yeah, even for it, we did not have many animals, definitely. So firstly, the heritability estimate. So we ran an heritability estimation for each of the functional PC scores. We corrected it with we included in the model a fixed effect, which is the number of cats that influence the blood metabolite profile. We did not correct it for the farm effect because the functional PC score were already corrected. And yes, we used the RML method and Wombat, and we ran it with a Wombat package. So here is the heritability for the blood metabolites profiles. So we could see that there was a significant heritability for the meal level of glucose, NIFA, and urea. There was also significant heritability for the peak height of the glucose and PHB. And it was slightly heritable also for the before-after ratio of the glucose profile in the blood during the underfeeding challenge. I will not describe precisely all the results for the 48 scores for the milk, but we could see that we had definitely large standard error, but it seemed that it was significant, especially for the principal components that corresponded to the mean level of the milk metabolites here, and especially for the milk metabolite that corresponded to an intracellular energetic pathway and protein-related metabolites. And the peak height for intracellular metabolic pathway were also significant, but for the protein-related metabolites, it was more the before-after ratio that seems irritating. Okay, and finally, the GWAS for the blood metabolite profile. So far, we only have 150 volts for that. It should increase in a few months. So all the goats were genotype with 50K Illumina bed shape. We run an SMP control quality, and we use a single SMP regression mixed linear model with the GEMA software. Here is the PCA for the 50,000 SNPs for our goats, and the color corresponds to the line of the goat. So we could see that there was a distinction between the high and the low longevity line, but there was also a continuity. It was not a strong selection we made. And we had a significant peak for one component, one score for the blood glucose profile during the underfeeding challenge. It was on the chromosome 6, and it was significant to a genotype level with only a Bonferroni correction. And these components corresponding to the high of the peak of the glucose. So it's the principal component here. And when we compare the goats with the different genotypes on this SNP, there is a AG and GG goats, we can see that indeed the GG goats have a very high decrease of glucose during the underfeeding challenge, whereas the heterozygote goats have a mild decrease, and the AA goats have a low decrease of glucose during the challenge. It should be confined by new genotyping of animals, but so far it seems wrong. So we had all the goats with no profiles that were genotypes. So we could compare other traits between those genotypes of goats, but we showed no difference between lines, firstly, longevity line, survival, weight, morphology, milk yield, and milk metabolites. So, conclusion of this part is that there are several components of the milk and blood metabolite profile that were significantly irritable. And the blood glucose decrease during the underfeeding challenge seems determined by the gene with additive effect on chromosome 6. We can guess that maybe it's linked with insulin sensitivity mechanism. Okay, so concerning the perspective, the primary aim of this project was to find a non-invasive and applicable at large scale markers of resilience. So we showed that indeed several metabolites that were discriminating between clusters, to say that these metabolites have something to do with resilience and the way that the animal reacts to the feeding perturbation, are also irritable. So those most promising milk metabolite constituents are glucose, glucose 6-phosphate, BHB, cholesterol, malate, and galactose. But can we use them tomorrow on farm to select the more resilient goats? Maybe not tomorrow. Firstly, we are not able today to measure this concentration on farm. One possible way would be to implement mere spectra to estimate the concentration of those metabolites in milk. But so far, it's not possible. Maybe tomorrow. But there is also other kind of sensors that are more and more used on farm. There is, for example, ways to measure today BHB, LDH, and progesterone on farm every day. So why not implementing all the milk metabolites? And another question is, should we submit our goats to an underfeeding challenge to see how they react? It would be complicated. So one way to deal with that would be to have daily record and to detect after unrecorded environmental perturbation. So here is one way to do it. Not me who do it, but it's the milk yield of a cow, a given cow. And based on that, you can use different roughness penalty to estimate a non-unperturbated milk curve for a given cow. And when you record are very low compared to the unperturbated curves, you can say, okay, probably this day there was a perturbation. And based on that, you can work and tell which cow or goat responding the best to your perturbation. So as a conclusion of this thesis, we confirmed the possibility of the selection on functional longevity. We have a better understanding on the underlying mechanism of functional longevity. We have a new pipeline of analysis to deal with this type of data. And we've got a proof of concept for new biomarkers of resilience based on milk metabolites. So thank you for your attention, and happy to respond to your questions.
Thanks so much. Does anyone have any questions? And maybe if you can repeat the question as well for the people online.
Yeah. Okay.
I'll get that one.
If I understand it.
Yeah. Yeah. So anyone with any questions?
Thanks so much for that presentation. I have a question on you on the first part of the presentation, where you had resilience and longevity in the same. Now, from your definition of resilience, it's a short-term recovery.
Yeah.
Whilst longevity is longevity.
Yeah.
Were you surprised that in your second part, you actually found 3 groups of animals, in the sense that there are always some animals that even when there's a disturbance, they will not show it.
Sorry,
So when there's a disturbance,
yeah, okay.
there's a group of animals that will never show that. Whilst there are 2 groups that show or not show, if you get what I mean. So were you surprised that actually there was a 3rd group in your PCA that wasn't well defined?
Okay. Okay. Well, firstly, the first question about the short term and long term, maybe. It's true that the longevity is a long term data. However, to live longer, you have not to die many times during your life, not to die. So it can be seen as a cumulated consequence of short-term events. And it's how we understand the link between the resilience, which is short-term, with longevity, which is what happens with the cumulated consequences of good resilience. And then with the result of the second analysis, I did not have strong hypothesis on that. I was quite curious because on one hand, there is several articles who show that the animal who show the less are the ones who live longer, and what we show it. But it could be the opposite could happen. I think. I guess it depends on the type of measure you have. You could think that an animal who responds much to a given perturbation is also an animal who adapts much and reallocate properly its resources. Here, it's not what we've seen. It's more that the animals who don't feel it, or at least don't show it in our measure, are the best one. Why not?
Right. Okay. Any other questions in here? And online as well. Question? Yeah. It's about the 2 lines that you were speaking about. Like you have 2 herds, but you have 2 lines within those herds.
Yes.
And what I understood is that between the herds, you separated them based on the benefit that they were showing. But there were no actual new generation that were coming from the individual.
We did not separate a given herd. Actually, we had we had the goats in Inha, and half of those goats were mated with semen from very good bucks, and other with semen with very bad bucks, let's say. Yeah. And then they were bred they were mixed in the herd. I don't know if it was clear. Yeah. Okay.
So like, I was thinking like, for the experiment, did you take the kids? So the next generation, or did you take the generation that you actually built?
There is several generations. Most of the goats are the generation 1. So the kids of the goats we inseminated with the bucks. A part of those kids were again inseminated with the extreme semen, but most of the goats were only generation 1. It's also why we did not see a very strong effect of the high and low longevity line, I think. And it's probably one of the reasons I think we did not see any difference concerning the metabolic response between the goats, because it's not two very different genetic lines.
Thank you, question. I'll go first and then we'll go over here. So were you able to estimate any genetic correlations between the different traits that you looked at and with also longevity? And then kind of the second part would be, yeah, which would be easier to select on milk metabolites or longevity?
Running the GWAS on 150 goats were quite optimistic, so genetic correlation was too optimistic with the number of goats we had. So there was no genetic correlation that were made. One asset of running the selection of on milk metabolite rather than functional longevity. Firstly, you can have the milk metabolites early in life, whereas for the longevity, it's at the end. So it can be a little bit more efficient. And then another thing we can think of is that the functional longevity is a kind of black box. You can live long because of many different traits. And when you select for functional longevity, you don't exactly know for which traits you select for. And probably it depends on the environment. Whereas when you select for on the milk metabolites, probably you select for more pure traits that is closer for the physiology of the animal. So it can be also an asset for selecting on milk metabolites rather directly on longevity.
Question over here?
Yeah, actually, it's an extension to that question in the sense that the metabolite. So one of them that you identified as a good indicator for longevity is BHP, which technically comes from ketosis.
Yeah.
So BHP is a very good indicator of ketosis. Now, when that animal recovers from this ketotic phase, doesn't BHP disappear as well in the system? So the metabolites, are they really good indicators for longevity when actually they may also disappear after that problem?
Yeah, I think it's why we need a very daily recording to get this this perturbation and to have this profile. If you only have one single measure, as we did so far for ketosis, we cannot select for resilience. I think you need daily measure to get it, because indeed it will not stay long. If it did, it would not be made in resilience.
Great. Any other questions? If anyone online has any questions, if you can raise your hands or speak up.
There's a chat.
So there is someone has left a comment in the chat from Nick Wade. He says, super impressive. We have plans to generate a very similar and possibly larger data set in farm salmon. We should talk about that one.
Yeah, for sure. Thank you. Yeah.
All right. Okay, so I'm not seeing any more questions in the room or online. So I want to just say thank you again so much for presenting. It's really nice having you here.